Hi, everybody. We have Dr. Brent Christner with us here today. Very excited for our first ever Fireside chat. So thank you so much for being here with us to talk about your work and the very interesting questions that students had as they dove deeper into your corner of the world.
Hi, Angelica, and all students watching. I'm very happy to be here.
So I kind of just want to dive right in because there are several opuses that you've published that were so interesting to the students, and just your broader body of work is very out of the box and got many students really curious about what's out there. So one student described you as a microbiologist seal, sea, air, and land. I thought that was pretty extraordinary. Focusing, of course, on bacteria and water, ice, aerosols, spanning topics from recovering bacteria from ancient glaciers to examining bacterial ice nucleators and snowfall, modeling bacteria in wildflower plumes, and examining microbes that are almost in space. And collectively, the students felt that the complexity of the systems that you are investigating was really interesting. And they appreciated your interdisciplinary techniques that you have to put into place to be successful in this space, collaborations from AFM to spatio temporal modeling. So they were really excited about this. So I guess the first place we can start is just more generally about how you got interested in this area. Some students were surprised to find this was even in an active microbial niche area of research. So can you talk to us about how you became interested in this specific body of work?
Yes, it was, believe it or not part of a great plan, a grand plan, I should say. When I entered graduate school as a PhD student, I was absolutely convinced that the most important thing that I should be working on were thermophiles. And so I actually looked out labs and looked for programs where I could go and study thermophiles. What I thought at the time would be the most important things that I should be studying. It just so happens the lab that I eventually joined, which was very heavily involved in hypothermophile research, had just begun to initiate a project where they were looking at ancient ice cores and trying to understand how far you could go back in time and still find things alive in the ice. And when I heard that that was a possible PhD project, I jumped on it. And then thinking, well, what a great PhD project, but of course, I'll still have to eventually figure out what I'll do for the rest of my life. I got a postdoc in ice-related research, and also thinking, wow, what a great opportunity it was. I was able to connect my PhD research and my postdoctoral research. I couldn't possibly get a faculty position where I could continue getting this research supported and funded. And I have been lucky enough to do that. And nearly 30 years later, I'm still working in an ice-related field with many, many questions still to answer.
Absolutely. And I know you had mentioned, just piggybacking off of that, you mentioned that this one in Brevia, one-page paper that you published, I believe in 2008 in Science, ended up laying down the foundation for 2 programs of your research. That's pretty serendipitous in a way. Can you tell us a little bit, unpack that for us, and what you mean by it catalyzing these 2 programs for you?
Yeah, you're absolutely right about the serendipity of my avenue into that research. Studying organisms in ice cores from times in the past. The ice that's actually formed and then accumulates in glaciers into sequences that can be many and many hundreds of thousands of years old in the polar regions. Those are organisms that at a time in the past were blowing around in the atmosphere. So by actually going with depth in a glacier, what you're actually seeing are species that were distributed, blowing around at different times in the past. When I actually became established as somebody who was working on these materials, almost specifically for trying to understand how organisms were surviving in the ice, there were I had a group of collaborators approach me because they had been very interested in the possibility that a certain kind of bacterium could be involved in actually affecting ice formation in the atmosphere and therefore the formation of precipitation. And they thought that a great way to test this would be to look at an ice core archive, which not only records aerosols such as microbes that were around in the past, but it also provides information on amounts of precipitation. And I agreed and thought that that was a wonderful idea, but I asked the initial question, well, okay, before we start melting 100,000-year-old ice, how many of these things are blowing around in the atmosphere right now? And how many would you find in precipitation? I was actually surprised to find out that we didn't know the answer to that. So what we started to do was to just analyze precipitation. I was living in Montana at the time, so collecting snow is pretty easy. You look out, it's snowing, finish your coffee, go out and collect snow, and that was a project that we had no money to pursue at the time. And that data set and that small one-page paper happens to be the most cited work in all the publications I made, all self-funded, which is unusual and I'm very proud of. And as you mentioned, it was just the start of work that we've continued to not only try to understand better the influence of these organisms. In other words, are they really affecting rainfall? A question that we could say is still outstanding. We don't definitively know the answer to that. What role might that actually have in the organism's life cycle? Does the organism really cares that it makes it rain? Or by actually forming ice, is that a mechanism that allows it to parachute back to the surface where it really wants to be? And then finally, these products that actually affect the formation of ice, that affect the way that ice crystals form and recrystallize, have enormous biotechnological potential. And we now have new projects that are trying to bioprospect new compounds that microbes make that affect this ice structure.
And
I started
that
a conversation over a coffee, about 15 years ago.
amazing, right?
Yeah.
Never underestimate the power of a single cup of coffee or a conversation or
Agreed.
um and so that gets to a question that one student just very blatantly asked, and that is, how would you describe to somebody not in this field, not a microbiologist, how would you describe A, what you do, and B, what exactly is an ice nucleator, and how do they help catalyze the formation of ice in the atmosphere?
Right. Well, the first one, broadly speaking, I guess the reason that I became a scientist is I'm interested in knowing how nature works. And that means even though I have very specific interest as it appears from my research program, my interests are actually broad at understanding that problem. How I got into studying this very icy aspect, I've explained a little bit. It is some serendipity, and also finding that there are so many questions. I mentioned thermophiles initially. The people who study organisms that thrive at very high temperatures are about 50 years ahead of what we know of cold loving organisms. And so just realizing that there's such a gap there, and it represents an opportunity to come in and make a mark in something that we know very little about. What exactly is an ice nucleator? An ice nucleator can actually be just about any surface that water molecules can attach to and interact. If you do not have an ice nucleator, it's still possible to form ice, but it will only form at very low temperatures that approach minus 40 degrees. And that's a process known as homogeneous nucleation. However, there are very few places on Earth where you actually can have homogeneous nucleation because there are particles and surfaces everywhere, except really high in the atmosphere. An ice nucleator enables water to freeze at much warmer temperatures because of the way that it allows those water molecules to interact on a surface. Now, any particle, dust particle, a human skin cell, even E. coli, can serve as an ice nucleator. What is really special about the kinds of bacteria that we've been focused on is that the organisms that this phenotype is very well described in it, they possess a protein that they express and they display in their outer membrane. And that protein is homologous to no other protein that is known. It has no other function. And what it is known to do is to actually organize water molecules in a fashion that mimics the ice crystal lattice. So these, other than ice itself, these proteins happen to be the most efficient at catalyzing ice formation, which is known. Biology can do it better than anything that we know about. The question remains, why on earth would an organism actually evolve this?
Mm-hmm. And I think that also gets to some questions that they have and I have about what the potential, and I imagine some of the stakeholders that you were alluding to earlier, what's the potential for harnessing this, especially that specific protein that you spoke to? It's not homologous to anything else, any other functions, for strategies for, of course, there's weather modification, cloud seeding, especially in regions sensitive to changes in precipitation patterns, but thinking even more broadly about global warming and about climate change. Is there potential there? And what is the landscape? What are some of the unknowns and the things we need to answer before we can start planning a strategy for how to implement this or study its potential implications?
Yes, I mean, of course, if bacteria are involved in the process of precipitation formation, it opens avenues for us to actually attempt to affect and control that, for better or for worse, right? And in the kinds of desperate scenarios that are projected for some parts of the world, this could be something that is very strongly considered.
Yeah.
Weather modification is not something, is not a new concept. For many decades, there have been attempts to actually modify weather, doing things like seeding compounds like dry ice, like silver iodide into clouds. The Chinese government has a huge weather seeding program. The real challenge is that to make enough ice in a cloud to matter, you have to widely distribute these compounds in the atmosphere. That can be done via rockets, that can be done from aircraft, but essentially it's very difficult and extremely expensive to do that in a widespread manner, in a way that could be effective. One thing I did not mention is that sort of the poster child for ice nucleation, an organism that is known as Pseudomonas syringae. If you've heard of that organism before, it may not be because it is an ice nucleator, although that is one of the things it's famous for. It may be due to the fact that it's recognized as one of the most agriculturally relevant pathogens that affect the foods that we think are delicious. Of course, one of the, as many decades of research that have been focused on controlling this pathogen, of course, the ultimate goal has been to remove it as something that limits agricultural productivity. Now, if this is actually an organism that is contributing in some way to the formation of rainfall, is that actually the strategy we might want to take? I'll just leave that answer. I'll just leave that out there. I don't know what the answer to that question up, but it brings up a very interesting question of things that we consider, something that we consider a bad guy might not actually be a bad guy. It actually might be, it might do more good than bad. And like, if we knew more about this, is it actually acceptable for a farmer to lose 15% of their crops, knowing that the presence of this organism is actually helping it more than it hurts? We don't actually know that that's the case, but that's something that we might consider.
This starts to get into more philosophical
It
questions
does.
and less so scientific.
So let's
Absolutely.
imagine that we had that all figured out, and what we understood is that one of the best ways that we could increase rainfall would be to increase atmospheric concentrations of ice nucleators, right? And in particular, those from bacteria. Well, I mean, we could certainly try to distribute these bacteria into clouds from airplanes, but we come up against the same problem that I just described, right? We need to get like zillions of airplanes flying around distributing these to matter. So we need a mechanism to get these into the atmosphere that does not rely on that. And you may or may not know that in recent decades, the amount of land area that contains plants that we decide what they are, in other words, agriculture, actually exceeds the land area of natural plants. So most of the places on the globe right now where there are plants are things that we've decided they will be there because we plant them again because they're delicious, right? Let's imagine that we actually used crops that encourage the presence of these organisms on their leaf. Maybe it's even possible to do this in a way that you don't encourage the ones that make the plant sick, right? Now what you have as you plant a crop is you're also in some way seeding the atmosphere with particles that could be altruistic in the sense that they could help farmers downstream actually receive rain. Now, you notice I say that just because you plant a field with ice nucleators doesn't necessarily the nucleators would go straight up and help you. And in fact, what it could evolve into is this a scenario much like the way that watersheds are controlled in the West, right? Like in Colorado, if you want to like save a little water that's running off your roof, so you can water your own plants and you put a barrel under your downspout, that's illegal. That is not your water. That is water that it belongs to the rest of the watershed. So do we now enter a territory where we're controlling water in the atmosphere and we're thinking about water rights that in the same way we do in land? I don't know. Again, it just brings up some interesting challenges that are not just associated with the science end of geoengineering on scales that we may have to do in the future.
And I love that you mentioned that something that may be characterized as a bad guy, in essence, I think what you were describing is that our understanding of that bacterium is limited. So we believe that there are horrible consequences. But then we discovered that there are actually really great advantages to allowing it to grow or to thrive or potentially facilitating it. So not everything is the good, the bad, and the ugly, right? And
Yes.
it comes back to this concept of relative truths. So our understanding and our own biases can sometimes hurt us because there's so much that we still don't know. That's really
Yes, I would
interesting.
agree fully with that.
It's so interesting. And I can't my daughter's a big fan of Jurassic Park, and so I can't help but think about Ian Malcolm, that nature finds a way, right?
Yeah.
Kind of crossing into some of that territory here.
And
And the
it
students
also we're very focused on the microbes right now, but like it's quite possible the plants are really just controlling the microbes that they allow, they
interesting.
encourage, and that
Yeah.
I mean, think of what message do you want to send in the atmosphere if you're a plant and you're in a place that needs water. That might be, I mean, it does seem obvious that there could be a way in which natural selection could operate on a process like that.
Mm-hmm. Mm-hmm. And then something that the students and I found quite fascinating and counterintuitive is you have found that wildfires can wildland fires can actually be an atmospheric source of biological ice nucleators. So the students had a lot of questions about that. One person was wondering specifically, do you anticipate any differences in those that are derived from prescribed fires, such as those that you studied, versus wild fires where there might be uneven burning patterns, potentially lower temperatures in certain areas? Or do you think those are negligible? And related to that, another other students, several were wondering, could we use that potentially as a mechanism to improve crop security? What are your thoughts? There's a lot to
Yeah,
unpack there, but I find it mind blowing. It's so counterintuitive that
I
fire could
agree.
breed water, basically.
That was definitely a study that I was surprised by, and just sort of the how this happened is that back to we're talking about biology affecting weather, right? Nobody disagrees with the idea that biology has found a way to form ice at temperatures warmer than any other particle. Nobody disagrees with that. Nobody disagrees with the fact if you had enough of these bacteria in a cloud, it could actually freeze enough water to make a difference. But for those things to happen, you have to find mechanisms to get enough bacteria into these high altitude systems to matter, right? So what are the ways in which microorganisms could be aerosolized in large amounts that they could actually affect processes in the way that I'm describing? Is it possible that simply wind blowing and aerosolizing microbes off plant leaves could do so in certain conditions? It certainly is. There are these huge dust storms that occur in seasonal ways, and large plumes of dust from Africa and Asia are transported across the ocean to continental scale transport. We know that those are particles that carry with them the kinds of compounds that could freeze ice, some of which appear to be biological in origin. Now, there probably isn't a more efficient way to mobilize material in the atmosphere than combustion. But then, of course, if we're thinking about things that are microbes that are alive, the idea that you're going to burn something to aerosolize them, it doesn't make a lot of intuitive sense, right? Because won't the fire burn them up? Which is what, of course, I would have believed. And in fact, when we entered the research, what I thought might be the case is that we could find that there was a lot of biological material aerosolized, that much of that biological material could serve as an ice nucleating particle. But I thought most of the organisms would be dead, right? So if we're thinking about a biogenic particle that can affect ice formation in the atmosphere, you don't have to have the organism alive to do that. But as a microbiologist, right, if you want to connect that to dispersal and the life cycle of these organisms, dead organisms, they don't work into that equation, right? So I will have to tell you that I was absolutely surprised that when we used different methods to assess how many of the organisms are alive in the samples. When we looked at aerosols immediately before the fire, since they're prescribed fires, we can actually sample air a minute before they initiate the fire. We're sampling air right before the fire and during it. And in the case of burning, you have a lot more bacteria that are in the air, and that can be for reasons separate from just being aerosolized by the fire, and I can go into that. But the fraction of bacteria that were viable, that we could find no significant differences between the two. So we couldn't even measure an effect that biology was having. I mean, we surmised that the reason that most of these bacteria are still living is that the particles that are in the smoke, many of them are, I mean, you can view them under the microscope. They are incomplete combusted pieces of biofilms that have come off of plant leaves, of what apparently would be chunks of plant matter, in some cases decaying. We even, when we stained cells on these, what you see are these big particles, and they can have hundreds of cells on them. These are like life rafts that are carrying these microbes up into the atmosphere. You've got lots of updrafts. So not really surprised that we were able to show that these fires mobilize a lot of stuff. Very surprised to find that not only is a large fraction of this microbial origin, but much of it is still alive.
Yeah, and I think, and it's interesting. One of our students even mentioned she's from Southern Arizona, so she really liked this idea potentially being able to utilize microbes or their proteins into the atmosphere above deserts where
Yeah,
she lives to increase
that's right.
rainfall.
Now, I'm just remembering the second part of that question. Do we actually expect there to be differences between
Right,
prescribed fires and
right.
more wildfires? And there's really good reason to think that there could be. Prescribed fires, meaning these are fires that are probably set every 1 or 2 years.
Right.
And that means low intensity fires. And those were the kinds of fires that we initially studied. So when we saw a lot of things alive, we often wondered if it had to do with the fact that these are essentially, they're not little campfires, but compared to the kinds of fires that occur out west, they're very different. And this work has actually been extended into a recent project that's involved not only looking at prairie fires in the Midwest, but also looking at crown fires in Utah. These are very high intensity, large fires, probably mobilizing an order of magnitude more material into the atmosphere. And what we're finding is not really in conflict with results from we're still finding very high concentrations of cells in these. So it's not necessarily that you get a fire hot enough, it will sort of eliminate the signal.
Okay.
There's still many, many, many microbes mobilized.
That's fascinating. And along those lines, you mentioned Utah, and obviously your research extends into many different places around the world. We had a lot of questions regarding how much experimental design goes into each project, and with having to deal with conditions in Antarctica, and then you were mentioning here some projects ongoing with wildfires. Students were wondering if you had any advice on designing and ensuring time on location is spent efficiently, especially it must be very expensive going to Antarctica, and getting not just statistically significant data, but data that you can interpret, dealing with confounds that you might not have anticipated, conditions that are outside of your control. How do you address all of that with your years of experience and working in these extreme locations?
Yeah. So logistically, in terms of how do you do this kind of research in a place like Antarctica? How do you do this hauling students there who've never maybe even know, who don't even own a winter coat, right? Like
Right.
this is Florida, right? Most of my students don't even know what snow looks like. And what I might say is that what I've been able to take advantage of is when I was a young scientist, I was paired with scientists who are now at my stage, who've had the experience to let me know that essentially in this kind of research, not only do you have to have every minute plan, you have to have every reagent, every article, every pipette tip must be accounted before you even leave. There's no Fisher orders that can be made from the middle of the West Antarctic ice sheet. Experimental design is also kind of quite often you must compromise beyond what would be the most optimal scenario. I'll give you an example. We've been involved in two drilling projects which are projects that required anywhere from $20 to $30 million each to fund, right? So these are sort of flagship projects from the NSF in its Office of Polar Programs. If you were studying a lake in any other scenario, you would go to that lake maybe, I mean, lakes change based on season. Lakes change based on position that you sample them in the lake and based on their depth, right? If you were going to do a PhD on a lake that exists in Florida, you might visit your site 40 times. You might have thousands of samples and observations from that lake. The last drilling project we had, when we drilled the little more than 1 kilometer hole into the lake, we had 8 days to do all the science. And what that means is that is probably on the order of about 12 PIs and about twice as many instruments having time to go up and down that borehole. In one place, you were not able to, I really like to sample both basins, or I'd like to do a transect to the coast. We have one hole. And through the one advantage that we have is that the ice that overlies this lake is actually moving. It's moving half a meter every day. So the one thing that we can do is actually get a sediment transect. We pull up a sediment core, the ice moves through the same hole, we can go into a separate place. All these things play into it, because even I'm giving you good news about the outcomes of that 8 days of 24-hour borehole time. But what would have happened if on the first day we would have stuck an instrument in the borehole? We didn't have enough fuel to drill another hole. We would probably have a dozen graduate students sitting there trying to figure out what they would do for a PhD. And I'll just give you a short answer on how we deal this kind of thing. We actually prioritize the measurements we make based on the risk that they incur to the borehole. So if we're going to put a Niskin bottle down into the lake and pull it up, the only real risk that we encounter is that we might lose the water, or it might freeze closed, and we might also not get a sample. If we're going to put down a 2,000-pound gravity core that's going to be jammed down in the mud, and we might not make it out, that could end the season right there. So we plan contingencies like, all right, if we had 10 liters of water and 1 meter of mud, what science could we accomplish? So plans on top of plans for things that may not ever happen are kind of the norm in research in Antarctica, where it's 3 times harder to do anything.
Well, and I'm just curious, thinking logistically about how that could be managed on teams of people. And some of these teams, everybody's there for a similar purpose and an overarching mission, but each team has their own unique needs and questions that they want to address within their grant mechanisms, right? And the reasons that they're there and what they have to deliver to their stakeholders. So I'm just, as you're talking, I'm thinking logistically, that sounds like a very interesting inner dialogue with a lot of planning and mapping things out. And my high risk, high reward might go out the window, like the timing of it, but then having to communicate that to other people and convincing them and yourself that that's the right plan of action, and adapting when they have other competing interests and input. I mean, it's just extraordinary.
And listening.
So how do you, it's the listening, right? So that's what I was going to ask, what advice you would have for people if they're ever in a position like this where they get the privilege to work on these, large teams of people. what is the number one piece of advice you have for doing something like this and making it successful? I mean, these are high stakes decisions that are
Yeah,
made, right?
it would be the same advice I would give for maintaining any relationship, and you just
Mm-hmm.
said it. It's communication. And what I really
Mm-hmm.
mean by that, first of all, it means being able to communicate your science in a way that scientifically literate people who may work in other disciplines can relate to it, and not
Yes.
only relate to it, but see how their science fits into it.
I
Um,
love that.
this
Yes.
cannot be a one-way street, though. It also means that you have to take extra effort to understand science that you may not be trained in, um, to leave your comfort zone. Um, and I've found that I'm not really sure that I feel like I had mentors that embed that in me very early on. So it's not like I became like an assistant professor and all of a sudden tried to figure out how to do interdisciplinary research. I was lucky enough to have a path that actually required that
Mm.
I mean, my two of my PhD advisors, they're ice core paleoclimatologists. They don't know anything about microbes. I had to learn very early to talk with them in a way that they know what I'm talking about. Um, so communication. Also part of this is it's just extreme luck.
Mm.
I could have described horror experiences with you being on teams where everyone fought and nobody communicated and nothing turned out like it should. And I've just been lucky enough to be part of projects where everyone sees the larger goal. People are altruistic about the way in which um, in which they do their science, are altruistic with their time. And I've just been super fortunate enough to have fantastic people to work with, people who are, some of my closest friends, people I would take a bullet for.
Mm-hmm. So I cling on to that word altruism. So we're talking about altruistic bacteria potentially, and then altruistic people studying these altruistic bacteria.
Yeah.
That's gosh, as we said, it just very quickly turns into this like a broader question of nature and philosophy, and it's so fascinating. I'm wondering too, on a little bit of a tangent, but how do you keep yourself healthy before you embark on such rigorous field work? I mean, I've been in situations before where a colleague had near heat stroke going out into the field in North Carolina in the summer, and just wasn't properly hydrated, wasn't used to the conditions. So thinking about the extreme environments that you're studying, what are some things that you do, principles that you might partake in right before leaving or when you're there, or what has kind of spilled over into your general lifestyle? What have you found that works for you to stay healthy, given all this demanding field work?
Yeah, well, I won't say that the field work isn't demanding. It doesn't necessarily require an endurance athlete to carry out. I would say as a young researcher, I probably just lived my life and didn't really think much about it. But as someone who's now in their mid 50s, who goes out in the field now with a bunch of 20-year-olds, that if I didn't do anything, I would get my ass handed to me by them. Uh, I I have found that the best regime for me, the daily exercise that keeps me at least from falling behind from these 20 years olds is I don't drive to work anymore. I ride my bike,
Nice.
and if I do nothing else in the day, I get 10 miles on my bike, and I ride it like it's a
Amazing.
time trial.
Yep.
So I think that is probably sort of my main route of not only burning anxiety and angst, but also of, keeping fit enough to, you pushing around a mouse and tapping on a keyboard isn't enough
Yep.
to
Yep.
allow you to haul around 90-pound packs for a few months.
So
But
I don't
making
know
staying active do.
this student I opened up, and it wasn't entirely a joke about being like a microbiologist seal. you're talking about laying out all the equipment ahead of time. That's like the manifest, right? Having to lay everything out in the military. And so as you're talking, I do see a lot of these elements. It's pretty extraordinary.
Yeah.
And another series of students were interested in work-life balance. And I think you kind of just spoke to that, where you've indicated you've consciously made commitments to yourself or to others that this is something you're going to do consistently for the end reward of, like, for instance, with biking every day to and fro work, the end reward is that you feel you're better equipped to handle these kinds of trips. So for me, I feel like part of the answer to that question would be these consistent commitments that you're making, and they're very intentional. But is there anything else, other advice that you'd like to speak to about your work-life balance and how you're able to do all of this?
Well, I mean, I would like to think that it's balanced, I employ some balance. This is the kind of job, it isn't a 9 to 5, 40-hour a week. You could run yourself to death. I guess there are probably some times when we're talking about investing 60, 70-hour weeks. I don't think those are uncommon. But I can also tell you there may be 3 days or 4 days that I might not do any work. For example, last week I was in Colorado for 3 days skiing. And that renews me. So I feel like this is a difficult subject. Some people don't like the idea of ever taking their foot off of the pedal. And those might be people that need that. For some people, work-life balance is like, they don't want to hear about it. To me, I think I don't know that I can enjoy the science and the progress we've made and enjoy watching my students go out in the world if I also wasn't enjoying my own life. So.
And what as I'm listening to you, I'm remembering early on in this series, you mentioned in this talk, you mentioned just having a general curiosity for how things work in nature. And I feel like a lot of that listening and observing comes from places of quietude, right? Or just being there with your thoughts and seeing what's happening around us. So I don't know. Now we're getting into Thoreau.
It is, and also those are thoughts that you can actually also have when you're on a bike or playing volleyball or at the beach, right? Like, this is a job that you can actually be still working when you're, and that can be bad in some ways, but
restorative in others, right?
Yes, I agree.
Yes. Wow. So I would think, some students were wondering about advice you would have for them. I feel like we just spent a lot of time talking about that in a roundabout way. But I really like this question, and that is, what's the best piece of advice you ever received, and how do you apply it to your life, be it personal or professional or a blend of the two? Especially given that you had such you had mentors who really, really resonated with you and helped you develop this language and this vision for how you interact with others and with the world.
Yeah, what would be the best piece of advice I've certainly gotten a lot of advice from my mentors. I might say that the best advice I've gotten might not have been from my mentor. It was probably from my dad when he was still alive, and it was like, always do your best. If you're going to do something, do it right. And I've tried to follow that. Sometimes that has meant incredible persistence. And if I have a strength, it's probably that: hard-nosed and persistent, not give up on I can have very little patience with certain things. But when it comes to, you find this as you start mentoring students, right? Most of your students come to you and they're usually not coming to you telling you about, oh, this is great and it worked. They're coming to you because I can't get this to work. I got to figure this out. This is a main part of my job, figuring out like how to solve problems. So I guess I might say that's probably the best advice I've gotten.
It's excellent advice. And I love it because it's simple, but the simplest things are really packed. There's a lot that goes into doing your best and persisting. And again, as I mentioned, I can't help but think of the correlates, the parallels with the bacteria who are surviving in these harsh conditions. They're trying to do their best too. It's interesting. There was one more question before I ask the last wrap up questions, although we could go on for for quite some time. There was a question one student wrote quite a series of questions for you, and it speaks, I think, to the downstream consequences of change, right? So she was wondering about after atmospheric transport and precipitation, these bacteria that can be spread, in the way that you were describing, can they deposit into soil? And do we know how or in what way they could impact rhizosphere microbiome? Or she mentioned causing disease, but I would also offer, on the other hand, potentially helping create a more thriving environment for plants. And I think it speaks to what you mentioned earlier about potentially plants being the controllers here. But what thoughts do you have on that?
Yeah, these are. I mean, most of the organisms we're talking about, right? These are things that are probably no more than 1 or 2 microns. If a microbe can actually survive the processes of aerosolation and transport into the atmosphere, for that organism, it's like a freeway transport system. A microbe, if aerosolized, could actually circumvent the globe in a period of a week or so. Apart from distribution in the ocean and the atmosphere, it's very difficult to think about mechanisms that would readily exchange gene pools and transport organisms over those kinds of distances. So for that reason, in trying to understand connections between gene flow and transport, I think the atmosphere is a very obvious. I mean, we thinking are very much in the outdoor atmosphere. COVID taught us about how important the atmosphere is I mean, COVID, although very effective at transporting in closed spaces within air, if it was really hardy, and it could distribute widely, we'd have a much larger problem on our hands. What are the implications of things that could blow around? We've talked a little about wildfire. We know that from other research that we've not been involved in, there have been previous studies that have actually implicated transfer of pathogens. In many countries, if a field comes down in disease, one of the practices is actually to burn the field. The idea being that this sort of eliminates the issue. We've not really worked in those systems, but obviously, when you look at the work we've done, it raises the question whether or not that's a really good idea. Are you actually mobilizing and distributing the pathogen? What we know from the increased frequency of wildfires in the West is that these communities that are very close to the fires, as well as the firemen who must be very close to the fires to actually fight them, there have actually been a few fungal diseases which have been implicated that it would appear the spores of these fungi are actually being distributed in the smoke. So in terms of wildfire, we can think of some examples that already exist that could suggest they could be blowing things around. In terms of back to the thinking of just sort of ice nucleation, I mean, an organism that is in the atmosphere, as I mentioned, is a very effective way to disperse. But the problem is that the atmosphere is deadly. Anything that is in the atmosphere is dying. Some things are better than others at actually persisting under those conditions. An organism like Pseudomonas syringae, if you put it in the lab and you did things like took water away from it to see how it responded to water loss, or hit it with huge amounts of UV, you would not find it to be extraordinary intolerance to either of those stresses. So on the surface, it looks like an organism. We know it's in the atmosphere. It's not necessarily the kind of thing that if you're going to build an organism to survive long-term, and it wouldn't be Pseudomonas syringae. But one of the features that it has, again, this potential way to pull a parachute so that it can be transported back down to the surface in a water droplet at some frequency, those cells land on a new plant leaf. And that organism could colonize the plant leaf and enumerate, and then be re-aerosolized, and this cycle could continue. I know that I've talked like this is a phenotype that is only harbored by one species. That's actually not the case. There are a number of other organisms that don't make plants sick that have this phenotype. And we have a project right now that's trying to understand a larger group of organisms that we think that do this. No surprise, when we normally use techniques that don't require that we culture organisms to detect them, we find new worlds of organisms that we've never described before. And my instincts is that is also going to be the case in the world of ice nucleation, and that Pseudomonas syringae may just be one of many microbial types, and not just microbes. There are certain fungi that are known to have this phenotype. There are plant pollen spores that are actually ice nucleation active. Again, interesting that a propagule that a plant purposely releases in the atmosphere can also do things like affect ice formation.
Absolutely. And I was just curious, you had mentioned to me another conversation about how you've learned that to study extreme environments, you don't necessarily have to go as far as Antarctica. And I just loved what you said there, the ethos behind that. And I was wondering if you might just expand on that for a moment. And we've talked a lot about the implications on land, but I'm wondering what complementary questions or projects could be pursued in the oceans, for instance, that might help further understand the implications of your work.
Yeah, no, you don't always have to travel a week to go to a place where you might find an extreme environment, like Antarctica. In Florida, believe it or not, we've studied icy systems for quite a long time. It's just that those are not systems that are on the surface. They're higher altitudes. It's interesting, if you wanted to study conditions on the surface of Mars, we're talking about logistics that are probably 1,000 times harder than actually studying something in Antarctica. However, if you took an organism and you put it at 30 kilometers in Earth's atmosphere, it wouldn't even know the difference. It wouldn't know the difference between there and the surface of Mars in terms of the temperature, in terms of the UV flux, in terms of the pressure. And so these are ways in which we can use places on the Earth to learn more about, about systems that are much more extreme than anything we might even find on the surface. As far as the ocean, the ocean is unlimited potential for learning about the microbial world. Not only because it represents the most volume of where life is on our planet, but 90% of the ocean is actually cold. Most of the ocean are deep waters below that are just a few degrees above the freezing point. And if you haven't realized it, what it really means is that most life on this planet is not in the sort of like warm. Most life on this planet lives in places that are cold.
Mm-hmm.
Which is fun if you've never thought of that before.
Yes.
And the only reason
Absolutely.
I didn't become a marine microbiologist is that I get terribly seasick.
Oh, no kidding.
I don't get seasick on ice. I had
Wow.
I had visions of me, puking constantly, like 3 decks below on a ship, like smelling diesel smoke and filtering for 12 hours. It was one of the main reasons I didn't go into marine microbiology, but
No kidding.
yeah, ice is good. Don't
Okay.
get sick on
Okay.
that.
So, I think, as I said, we could extend this conversation well beyond the hour, but I have two last questions for you that I definitely want to capture, and that is, if students are watching this and they're really excited about your work, um, what are the next steps for them? What should they be doing if they're interested in what you're doing? Do you have a mechanism for our students to reach out to you, or do you have advice for them if they're applying to our PhD program, for instance, on how to move forward with that if they're interested in your work? What advice would you give them? And related to that, what do you think? I can kind of extrapolate what you believe to be one of the greatest challenges facing microbiologists today, because you're trying to tackle that. But related to that, what would be one of the greatest challenges that a budding microbiologist might face as they look forward into a potential career in this area? So sorry, that was
Okay.
kind of part A, part B, but I know we have
Yeah.
some students who are very excited by your work. So I want to make sure they have a path forward if they're interested in this
As
area.
as far as a path forward in interest in my research, sending me an email is probably the best way to do that. As far as a pathway into PhD research, I might say that there are so many opportunities now to engage in the kind of research that connects different disciplines, what we call interdisciplinary research. I might say if somebody was interested in what we do, but doesn't work with us, that the kinds of evidence that you can find for that are people who study and publish in journals that are not necessarily all microbiology journals. That is maybe a
Absolutely.
really good
Absolutely.
indication of someone who's not afraid to step outside
Mm,
of discipline boundaries. Uh, as far as what might be some of the greatest challenges facing a student in micro now, all right, here's sort of, I'm kind of copping out on this one. I might say there are so many interesting things to study in the microbial world. Actually, choosing the right one or
mm, mhm.
choosing one. Uh, I can tell you that if I would have stayed the route of studying thermophiles, I feel like it would have been very rewarding, and they're just incredibly interesting adaptations which are involved. But I also believed I'd probably be fighting with dozens and dozens and dozens of other labs
Mhm.
that are all sort of tackling a similar problem.
Mhm.
And I might say that the benefit that I share now in the area that I'm in is that is that almost everything we learn is new, and that the kind of fear of being scooped is the kind of work we do tends to keep the competition down. Whether that be because to do it you got to be in Antarctica, or like sail a balloon at 20 kilometer. I don't know that has to be the answer to how you design what research you do, but I might say that new avenues are not just shiny; they're opportunities
Mm,
to really fill
mhm,
gaps and make your mark.
mm.
And that's what I've tried to do.
Excellent. Excellent. Well, gosh, we've been able to ask so many incredible questions and really cover your work. The final question is, what question might you pose to our students? Just something to end on. It could be a rhetorical question, but something that you think. You mentioned that listening is so important in this field, and you've also highlighted the need to to have to restore and to spend time alone and think. So I wonder, while students are doing this, what is a good question that you think they should ponder and might get some benefit out of?
Good question. I don't know. I mean, I'll go with something. I'll go with something simple. How does it work? Um,
Yeah.
you could really apply that a question to anything you don't understand, and that might be. I mean, you talked earlier about curiosity. Where does that come from? And I feel like the little voice in my head every time I learn about something or exposed to something that I don't really understand, that's what the little voice
Yeah.
in my head says. How did it work? So I might say, if that's a really great question to ask if you can't think of any other.
I love it. I love it. Well, on that note, thank you so much for your extraordinarily generous time today, for sharing so many different corners of your world with us. And I hope that all of the students listening really enjoy this talk. Maybe it sparks something for you. And thank you so much for your time. This was really fun for me.
Well, thank you, Angelica. And I wanted to also thank the students. I know that there were so many questions
Yeah,
we weren't able to get to, but I had so much fun reading through them. Some were so well thought out that they were great.
thank you so much. And with that, we close our first iteration of a fireside chat with a microbiologist. Thank you, Brad.